Hi, I'm Sebastian Thuault. I'm a senior editor at Nature Neuroscience, and I'm here at the Cold Spring Harbor Laboratory for a conference on brains and behaviors, order and disorder in the nervous system. And sitting next to me is Helen Mayberg from the Icahn School of Medicine at Mount Sinai in New York City. Helen is a renowned neurologist working on depression who is attending the meeting, and she talked about deep brain stimulation and depression during the morning session. So Helen, before we go into the details of your work, I wanted to know if you could tell us a little bit about the condition, the disease. We all know individuals with depression, and I wanted you to do perhaps a little bit of a presentation on what's this condition, what are the therapies that are available to this patient population at this time.
So depression is a very common psychiatric condition, one of the most common that we have. It's diagnosed by an interview, not by a lab test or a scan, but it's a condition that's characterized by a disturbance in one's mood, generally a profoundly and distressing low mood that's accompanied in most cases by a sense of not feeling pleasure. Anhedonia. And then it has an admixture of many other symptoms that affect our drives, like sleep or appetite or one's libido, activities, as well as interests and thought. So thinking can get slow or confused. People feel like doing less. They may feel anxiety. They may have sensations in the body, but at its core is a disturbance in drive states, emotion, and putting thought to action.
And so in terms of drugs or therapies that are available for this patient population at this point?
So despite being extremely common and accounting for much of the world's disability, remarkably at this point, the good news is that there are many treatments and have been. So there are a number of evidence-based psychotherapies, like interpersonal psychotherapy or cognitive behavioral therapy that can be quite effective. There's a multitude of medications and have been since the late 50s, kind of heralded now by the serotonin reuptake inhibitors, but also drugs like tricyclics or mixed serotonin and norepinephrine drugs. We have new medications like ketamine that are being tested that are not even of the old classes. And we have somatic treatments like using magnetic therapies, TMS, but most importantly, the best treatment that there is actually electroconvulsive therapy, but it's generally reserved for people who are extremely ill and have not responded to medication or psychotherapy.
So in your line of work, you've actually explored another approach to therapy, which is deep brain stimulation. And from what I understand, this is something that was inspired by the use of this approach for motor disorders like Parkinson's disease. And so why don't you tell us a little bit about your research in this area, perhaps starting by how did you decide initially to explore this strategy, this avenue to provide patients with some relief?
So in the old days, I was a neurologist that started my research as a postdoc in our radiology department, and I had the opportunity to work on chemical mapping with PET scanning. And I actually studied epilepsy in my first experiments, but I was introduced to a team that was studying depression and neurological disease. And we started to apply the tools of imaging to those depressed patients because we had a lot of internal controls by disease, and started to map that there were consistent abnormalities across different groups of depressed patients. And when we had some confidence that there was a signal from the scans, we moved to use conventional treatments to treat people and to look to see how brain scan patterns corrected. We fell on this region that actually wasn't abnormal to start in our less ill patients, but it downregulated its metabolism when people successfully recovered with medication. And we saw the same pattern with placebo responders, and we tried other medicines, and then we saw the dynamics of this region even with psychotherapy. And we started to kind of hone in that many changes in the brain were happening, and we were starting to fill in this map, this sort of pseudo circuit, macro circuit of depression. But this subcallosal cingulate area 25, Brodmann characterization, seemed to be vitally important, and that if it didn't change, you didn't get well. So we had this over many, many years, this evolving mood circuit. We're looking at these macro dynamics with imaging over, months. And when I moved to Toronto, I was introduced to some neurosurgeons and was also working with the Parkinson's group. And so the field in Parkinson's had, for several years, been working on tuning basal ganglia circuits with the work that we're all now very familiar with, but that they had a motor circuit, and we had actually modeled a lot of our original depression analyses on basal ganglia circuitry. So I kind of revisited the basal ganglia story. It was like, well, they have a circuit. I mean, they happen to have it very much more worked out than we did, but we had a circuit. We knew we wanted activity to be attenuated. We knew if you couldn't talk it or drug it or shock it into normal functioning, maybe we should leverage this technology that they were doing with precise placement that we could implant, we could stimulate, we could use high frequency, which in their hands was blocking an aberrant signal.
Okay. Which fitted with your hypothesis that
so
we needed to...
at that time, it fit our hypothesis. It turned out it's the wrong hypothesis, but the good news is that we had a clinical signal.
Okay, and so can you tell us a little bit about this particular technique of inserting electrodes and how local is the stimulation, what it does to the area and perhaps beyond the area?
Yeah. So this procedure is a standard neurosurgical procedure. It involves being in an operating room. A neurosurgeon will make a small opening in your skull, about the size of a nickel, one on each side, right at the top of your head. And then you are placed in a stereotactic frame that allows communication to go between your MRI scan, where you can see a person's anatomy in 3D, and a coordination and navigation system that allows the neurosurgeon to translate where we see on the image to how he can plan a trajectory and placement of what turns out to be a small wire. It's about the size of a strand of vermicelli. It's about a millimeter and a half in diameter. And that wire is placed under sterile guidance with the patient awake through a cannula to a designated location in the brain. So it's placing of a wire in an open way into a human brain.
And so this is an awake surgery?
It can be done awake or asleep, but by having patients awake with Parkinson's disease, you're able to record and to know when you reach the target because of the characteristic physiological firing patterns of parts of the subthalamic nucleus or adjacent areas. So physiologists have learned what the signal is for basal ganglia implants. We wanted patients awake so that we could record and see if we also had some localizing physiological signals. Turns out there wasn't anything nearly as helpful or really helpful at all. But we, with the imaging guidance and information we had from our prior studies, we were able to make a reasonable approximation of the target. And so that was, we used, once we guided the wire to where we wanted it to be, we used behavior, actually, to be really transparent about it. We didn't know what was going to happen. Part of studying the initial patients awake was because it could have gone exactly the opposite of what we wanted. So, but by having patients awake, we could turn it on, we could test different parameters. We really weren't ever expecting anything to happen immediately.
And so I mean, you collaborate with a neurosurgeon, you know, for these approaches. Are you or were you in the room, you know, the operating room with the team during these first experiments?
You think I would miss that?
So why don't you tell us about, you know, perhaps the first time, you know,
you know, the first time is always the most memorable of all events, but it's also the most nerve-wracking, because at a point you go from an idea to getting all the permissions to actually finding a patient that's agreeable to then moving to an operating room. You move from kind of the thought experiment to reality. And so the patient was awake, and once the implants were done, which was quite straightforward, I mean, Dr. Lozano is a very experienced functional neurosurgeon, that the patient was basically prepared that this is a point that your job is to basically tell us if you know anything, because our intent was to just make sure that giving current in this location was safe. For me, knowing the intricacies of the neuroanatomy of this location, I was concerned that we would have some instability in blood pressure or pulse or kind of drive functions because of the connections to the hypothalamus and the insula. And so everyone was poised, because all you do is when you start with low current, you go to turn it up, you're just watching. So you have monitors, but basically you have the patient who, because the patient wants to get better, but on the other hand, doesn't know what's going to happen, and we don't know what's going to happen. The instruction is, you just need to tell me if you notice anything, period. Good, bad, indifferent, or nothing. There's no right answer. So you start, the electrode has got 4 places you can stimulate. Makes sense to start at the lowest one with the lowest current and just slowly turn it up, and nothing happens. So we move to the next contact. Start at the low current, turn it up, nothing happens. Kind of going, okay, keep going. Go to the third contact, turn it on, turn it up, and we get to about an intermediate amount of current, and the patient goes, "Did you just do something?" And I was like, "Well, why do you ask?" And he goes, "Well, I suddenly feel incredibly calm." I said, "Well, maybe just getting more relaxed." She goes, "No, what are you doing?" We turned it up a little more, and she goes, "Well, that's interesting, the void just lifted." And I kind of went, "What's a void?" You know, not a term that's in the psychiatric nomenclature, and certainly not on the clinical checklist. And as she tried to describe what had happened to her, I mean, that was that moment when we realized that maybe there were things about depression that could happen very rapidly, not our expectation. We were in that OR to see that we didn't do anything bad. And she began to describe this calmness that she hadn't remembered in a long time. And then as she tried, then we kind of went into, is it like Valium? Are you falling asleep? You know, what are the choices of calm? And it was clear that this was some internal state where something very negative had just been released. And then we turned it off, and she said, "Oh, maybe I was just imagining it." And then we turned it on again, and it lifted again, and tried to get her to describe it. And she says, and she got very frustrated, and she goes, "You're trying to ask me to describe the difference between a laugh and a smile." And then as she tried, and we left it on and tried another contact, didn't get the same thing, she explained that it was like the first day of spring, and this was in Toronto, where you go outside, it's still freezing, there's snow on the ground, and you see a crocus popping through the snow. And you can just imagine that spring is coming. And she's describing this to us, and I'll tell you, that was in, you know, May of 2003, and I can still remember that sensation of being so worried something would go wrong, and this would be my fault for starting this, and then listening to a woman that literally had chains caught. It was so subtle. It was not laughter, it was not hypomania. It was this subtle that there was a sense of relief. And because we had, or not me, the movement disorder people routinely, when you got in the right spot, turned the current up, could watch a tremor, immediately halt. I think we all were wondering what we were watching, but we wanted to get the other electrode in, retest, and then get out of the operating room, figuring we would get similar phenomena if we did it outside the operating room. But that first case, and we did not always see that early on, but over time, that thing seen in that first patient has become a very, very critical part of the experiment.
So it sounds like it was really an amazing experience for you, also as a researcher, as a clinician. You say you remember this very well. So, and you know, you telling the story is really, you know, very interesting. So what happened after that? Because you tried to scale up your efforts and work with different cohorts, and you talked a little bit this morning about, you know, some of the clinical trials you've been involved with, and so why don't you tell us a little bit about the results of this, and perhaps we can talk also after that about what's next, you know, what the future holds.
So, you know, like any experiment, you know, it's not an N of 1. So you do 1, we did 6, published. We got 4 out of 6 better. We expanded to 20, got 60% better. And industry became interested in this may be something to develop commercially. They ran their own pilot studies and got not quite as good a result, but actually a good signal to be able to pilot a clinical trial. In the meantime, I moved from Toronto to Emory. I had a new team, new surgeon, new psychiatrist. We started to go independently, separate from both Toronto and the clinical trial, but we're very interested in mapping out where exactly we were and how to understand the circuit better, not just the spot. And so there was a divergence in a way where we were trying to do small trial experimentation and try things out to improve. Clinical trial going with what seemed like a decent signal, and the two studies actually diverged. So while we got much better and started to see that intraoperative effect in a predictable manner, could understand what was different between responders and non-responders by our imaging maps, and then could customize our targeting for individuals. Clinical trial went with the original method. So the Broaden study that was run and sponsored by St. Jude Medical Incorporated, a device company, basically set up a 6-month active versus sham comparison that at the halfway mark at 90 subjects didn't show a difference. And the company felt as though maybe they need to go back to the drawing board and look at why had that happened, because that was certainly not what they had projected or anyone had projected, and certainly wasn't our experience in Atlanta, wasn't the experience in Germany or in Spain or in Calgary where other groups were doing these small studies. So the interesting thing about the industry trial is that while they were focused on an endpoint for FDA approval at 6 months, patients continued to have chronic stim and did much better with longer stim. That was a failed trial, not a failed treatment. And that's sort of where we are right now. We have grant money to actually study with prototype devices, signals off the brain, but we have an arrested trial so that patients are in a limbo unless you're in an experimental study.
So one of the things that I understood from your talk this morning is that all this experience you've had with these patients, these cohorts, is you seem to think that you actually need to gather as much information as possible from them, and perhaps along different clinical dimensions. And so you were recently appointed as a new director of a new center at the Icahn School of Medicine, which is called the Advanced Circuit Therapeutics Center. So can you, perhaps to finish this conversation, can you tell us a little bit about what you're going to do, and perhaps what other, from which other angle you're going to approach this terrible disease?
So the opportunity I've been given at Mount Sinai is to build a new center. It's not from scratch, but it's to embed a research group right in neurosurgery with functional neurosurgery. And that's not a new concept because every functional neurosurgeon has got research collaborators. What we're going to do different is we're going to take the clinicians that get routine movement disorder surgeries for Parkinson's or dystonia or tremor, and those clinicians will be managing their patients in the same clinical space as the psychiatrists that are using DBS for OCD or doing experimental DBS for any disorder. So my research depression work will live alongside clinical movement disorder, and we will have shared platform for measuring mood, motor, behavior across diagnosis, because we're tuning circuits broadly. We're not treating specific syndromes or disorders. It's an artificial separation. And only by actually getting cooperativity from the institution and from the departments to say, we don't want to keep this in our own silo. We're going to move everyone together. I'll be able to look at non-motor features of Parkinson's where they treat the motor problems exquisitely well. They do nothing for apathy, emotional instability, OCD. And in depression, one of the most striking things that I find is that emotion locks down your ability to move. So there's actually more alike between a Parkinsonian patient and a really profoundly ill, depressed patient than we've thought of. And if we look at them experimentally through the same lens, it's scientifically efficient, but I think we can really learn from the syndromes because, facial expression in depression is profoundly abnormal. It is in movement disorder as well. Voice, speech, gait. And so we can share knowledge across diagnoses, I think, in a new way. And so when we get new devices, for Parkinson's, we can be thinking about how they might apply in psychiatry. And because now devices allow you to do more than one thing at the same time, maybe we can evolve to where we treat mood dysregulation in Parkinson's and motor off the same electrode. So we can develop technology innovations as well as clinical innovations, and we'll have partners together, and it'll be fun.
That sounds very exciting.
It'll be fun.
Helen, thank you very much for sharing your thoughts, your experience and ideas with us today. It was very interesting, and I look forward to the new development coming from this center.
Well, thanks for your interest. Thanks for your interest and for the opportunity.
Thank you.